Mr. Toastmaster, fellow Toastmasters, and welcome guests. There have been enormous advances in the development of means to help people with Parkinson's disease. I've had the good fortune to participate in many studies, and I have the great fortune to be working with an outstanding group of investigators for current and future studies. I would like to discuss with you some of the advances. We are going to be discussing the use of a medication that is not approved for general use. We are also going to be discussing the use of some products which are also not approved for general use with humans. I'd like to point out that the device that we're going to be discussing has been approved for use with humans by clinical engineering at Johns Hopkins Hospital, and additionally, it has been approved by the Institutional Review Board for use with participants. So we have the informed consent of all participants in this study. However, the drug and the device are not approved for general use. I have no financial conflicts to report. I'd like to go through my personal path, how I developed my interest in helping people with Parkinson's disease, going through the motivation for this project, the overall vision, the specific mission, core values that we have, and some of the ongoing past, current, and future studies that we're working on to advance the treatment of people with Parkinson's disease. Eventually, we aim to prevent and cure this disorder. I began my career in psychiatry by reading a book about psychoanalysis when I was in high school. So I developed an interest in understanding the mind. That led to my desire to become a psychiatrist. When I was in medical school, I was impressed by Dr. Norman Geschwind about the relationship between parts of the brain controlling movements and sensations and how interruptions of those pathways could lead to disorders in individuals. At that time, Dr. Geschwind pointed out that there likely would be parts of the brain associated with behavior and emotion, and that has come to fruition. So that led to my decision then to become a neurologist and psychiatrist. Therefore, after having training in internal medicine, I went on to complete training in neurology and psychiatry. Because I've had a special interest in the disorders that affect children, I pursued training in child and adolescent psychiatry. I then had the good fortune to participate as a fellow and eventually coordinator and program director of a research program in developmental disabilities through the Office for People with Developmental Disabilities of the state of New York. And after that, I then went on to study brain imaging. And for the past couple of decades, I've pursued training and have continued on the faculty to conduct translational imaging research in neuroimaging using positron emission tomography primarily to look at chemicals in the brain of people with nervous and mental problems with the means of helping tools to correct whatever abnormalities may take place. I am continuing my research career at Johns Hopkins, and I seek to continue collaboration with colleagues throughout the university, as well as at other institutions. So I'm eager to develop new imaging techniques, new translational research with colleagues around the world. The overall motivation for this current project is to look at the physiological basis of Parkinson's disease. The goal is to develop means to better diagnose the condition and to develop tools to treat, cure, and ultimately prevent the disorder. My vision is to develop equipment that will visualize the functioning of an individual during radiological and other procedures. So I'd like to be able to visualize the chemicals, the interactions within the brain, at the same time as measuring movements that take place in the body and making assessments of the psychological, behavioral, emotional aspects of the individual. So I'm imagining a huge apparatus that will combine current radiological, nuclear medical, electrophysiological, as well as visual measurements to advance the diagnosis and treatment of people with Parkinson's disease. The initial mission currently is to develop some tools to currently help people by means of improving diagnosis and treatment of people with Parkinson's disease. The core values would be to ensure the safety of the participants and patients and to develop equipment that's of optimal quality. Parkinson's disease is a disorder that is characterized by deterioration over time. It is caused by the death of the nerve cells that produce the chemical dopamine. Dopamine is a neurotransmitter. It is an agent that's present in healthy people. All of us have dopamine, and it is a chemical that stimulates nerve cells to produce the behavior that we need. In Parkinson's disease, the cells that produce the dopamine die, so there is a reduction in this substance. The causes of Parkinson's disease are many. There are genetic influences, there may be environmental influences, toxins, chemicals, and there are other unknown causes of this condition. Parkinson's disease is characterized by the presence, commonly, of a tremor. It's typically called a pill rolling tremor. So it would be a shaking movement. It may appear on one side more than the other. Additionally, people with Parkinson's disease have slowness of movement, stiffness of muscles, and this interferes with their posture so that it may be difficult for them to ambulate, and that may lead to falls. I've had the good fortune to take part in a number of studies of Parkinson's disease using positron emission tomography to look at the chemicals of the brain, as well as doing assessments of the individuals, the movements and behaviors of the individuals during the course of the investigation. In a study of the changes that take place during the development of Parkinson's disease, we noted that there was inflammation in the brain that occurred early in the course of the condition before symptoms developed in the individual. It was later when the symptoms developed that there was evidence of the death of the cells that produce dopamine. We also investigated the use of GM1 Ganglioside. This is a medication that has the potential to help people with Parkinson's disease. Another useful tool for people with Parkinson's disease is deep brain stimulation. This is a condition that involves the placement of some electrodes, some metal devices within the brain to stimulate the brain, and it is a tool that is useful for people who have not benefited from the medications that usually help people with Parkinson's disease. So during the course of a study of the effect of deep brain stimulation, we identified that there was improvement in the symptoms, motor, nervous, and a reduction of depression symptoms. At the same time, there was an increase in the amount of glucose, sugar, that went to the parts of the brain that are associated with motion and memory. For the past 4 years, I've been working with Dr. Timothy Harrigan, a research engineer at the Applied Physics Laboratory. We met during a translational meeting scheduled by my department and the Applied Physics Laboratory. And since then, we have developed a fruitful collaboration. So I expressed to Dr. Harrigan my frustration at doing movement ratings during scans over many years, and my recognition that with visual observation alone, I am missing some important features. There are limitations to what I can do just by visually observing people during typical ratings. I've always thought that there must be a better means to utilize technology to record the effects of various interventions on people with Parkinson's disease. And fortunately, Dr. Harrigan is extremely skilled putting things together with his hands, and he was able to put together some simple, inexpensive equipment to measure the movements of people with Parkinson's disease. We were interested in studying the effects of interventions on people, measuring their movements with instrumentation. At the same time, they were undergoing positron emission tomography and other radiological procedures. 4 years ago, we had the good fortune of having Gregory McKay participate with us. And Gregory developed a protocol to utilize the instrumentation developed by Dr. Harrigan. And Gregory was particularly interested in the improvement of the techniques to diagnose and treat Parkinson's disease. So typically the assessment had been based on a clinical assessment of symptoms and signs with a structured rating based on interview and examination. Gregory developed a means to improve this using instrumentation. Along with Dr. Harrigan, the use of an accelerometer was devised. This is a tool that provides the representation of the 3 dimensions in space, X, Y, and Z axis. This is a simple device that then can be placed on the extremities of individuals to give a representation of the position in space and to record changes that take place during time. So this is a simple device that can be placed on the extremities of people with Parkinson's disease in order to record their movements. The output that takes place is a linear representation. So typically, the accelerometers are placed on the finger and wrist of each upper extremity or on the toe and the ankle. Since there are 3 dimensions represented by each of the accelerometers, the 4 accelerometers for either the upper or lower extremities are represented by 12 lines on the output. This is a demonstration of the placement of the accelerometers on the upper extremity and on the lower extremity. This is a representation of the output that takes place. So with time that is represented along the horizontal axis, there are changes. And to assess this instrumentation, Gregory produced movements of varying degrees of severity. And the vertical axis shows the voltage that was generated in the accelerometers. And we can see that as the tremor increased, the change in position vertically increased. And this is a summary representation of this. In this way, along the horizontal axis, the number of movements per second are represented. And the vertical axis is a representation of the power or the force, the output that is generated with each of the different types of movements of varying severities. We can see that with the tremors that were generated by Gregory, there was a base frequency at about 6 cycles per second with some secondary harmonics at 12 and 18 cycles per second. So this is a representation of the way in which the output is being analyzed. In addition to performing the movements with the instrumentation, we utilized a visual observation that was modified from the Movement Disorder Society version of the Unified Parkinson's Disease Rating Scale. This is a well-established scale that is commonly used by neurologists for clinical and research purposes. So this is the gold standard by which people with Parkinson's disease and related conditions are assessed in the office. It involves performing multiple activities and scoring the observed behaviors from 0 for normal to 4 for severe. Fortunately, the Movement Disorder Society has kindly given me permission to utilize this tool and to modify it for our investigation. The movements are typically rated by looking at the amplitude of the movements and to look for involuntary changes that may take place during the movements that could hinder the performance of the movement. These could take place in the form of interruptions, slowing, or decreases in the movements. Additionally, when looking at the movements, we are interested to identify whether or not they take place on the left or the right side. This is a representation of the movements that are performed typically with the upper extremities. And then there are 5 movements that we commonly perform with the lower extremities. So far, we have assessed this protocol on a number of individuals. We have studied a single person with a form of atypical Parkinsonism, multiple system atrophy, the Parkinson variant. We've had the good fortune of having the participation of 20 people with Parkinson's disease. 10 of them have gone a single test session. 10 others have done both a test and retest session. We have also studied some healthy people, and 8 of these people have undergone both test and retest sessions. These output are being analyzed by means of transforms, and I had shown some representations of the way in which Fourier transforms are expressed. There are some additional, more sophisticated ways of presenting these, and we are currently proceeding with those analyses. So we are challenged by the current inability to access research laboratories to obtain our documentation. And there are activities going on that are being hindered by the health crisis. I've had the good fortune to have obtained the collaboration of an outstanding group of current and future scientists and physicians from around the world who are participating in the analysis of the study that we have already completed. These involve the use of tools to utilize the transforms and other sophisticated analytic tools to make sense of the data that we have corrected. And we are going to be presenting this information at the meetings in the near future, and we are in the process of presenting and publishing the output. So I am in the process of collaborating with a team of outstanding individuals from around the world to assess the information, and we are going to be soon presenting and publishing that output, those findings. I'd like to thank the sponsors for my research. I'd like to thank my collaborators, because I could not have done this without their participation. Additionally, I'd like to thank all of the participants who have participated in this. The research is expensive, and we are seeking grants to continue. We also welcome the participant of other donors.